what we're going to be talking about today is joint modeling of exercise response and circadian rhythm in RNA-seq data across 15 tissues. This was really a team effort led by Jun Lee and Chuck Arnt, and myself, Jiayu Zhang, and Yanling Zhang really put all this together. Okay, so here's today's talk in a nutshell. We're going to talk about the purpose and the design of the study. Really, the purpose of the study is to measure the acute exercise response. That was the original purpose. But then we ran into a problem with confounding. Specifically, circadian rhythm and circadian effects seem to be influencing gene expression. And so what we want to do is, in today's talk, we're going to create two models. We're going to model the acute exercise response. We're also going to model circadian rhythm. And then the ultimate solution is really to combine those models into one, where we have one part of that model measuring the acute exercise response and one measuring the circadian rhythm. So we're simultaneously modeling both of these effects in a joint model and computing them for their percent variance explained. Now, if that doesn't make sense right now, don't worry. We will walk through these steps one by one. So in the Lee lab, we work in MotorPack, and MotorPack stands for the molecular transducers of physical activity. And really what we're trying to do in MotorPack is figure out at the molecular level, cellular level, how the human body responds to exercise, whether that exercise be aerobic, whether it be anaerobic. And this is a multi-species study. We're working with humans, we're working with rats, and the rats help us. They give us tissues that, of course, humans can't. And this is also a multi-tissue study as well. So let's jump into one of our first studies, one of our first data sets. This is actually called the PASS phase 1A, or really we're just going to call it the PASS 1A study. And it aims to study the acute exercise response over time. So what we did here is we took 7 cohorts of rats to undergo acute exercise response. So they went through an acute exercise bout where they had to run 15 meters per minute at an incline. So it's a 5-degree incline for about 30 minutes. And that's what this red represents right here. This is that exercise time. And then afterward, there are 7 time points. Immediately post-exercise, 1 half, 1 hour, 4 hours, 7 hour, 24 hour, up to 48 hour, where these rats were sacrificed and their tissues harvested. We compared those to sedentary rats at 0 hour and 7 hours. So these sedentary rats, of course, are sedentary. They didn't undergo any exercise response. In total, if you were to add all these dots up, we have 78 rats, 15 tissues, and this is a multi-omic study. We're dealing with RNA-seq. This is bulk RNA-seq. It's not single-cell RNA-seq. We also have proteomics, metabolomics, and bisulfite sequencing. So let's jump into the data inventory real quick. Here are those 15 tissues lined up, and they're lined up by their sequencing batches. You can see there are 5 sequencing batches from 2 locations. MSSM stands for Mount Sinai. So there are 3 batches, sequencing batches out of Mount Sinai. And then there were 2 out of Stanford. And in each of these tissues and batch specific cohorts, what we can see is that 78 samples were sequenced and processed at a time, with the exception of ovaries and testes, and that's because ovaries and testes are sex specific. So, although we have 78 rats, given that we have 15 tissues in the RNA-seq, we end up with just shy of 1,100 samples. Now let's just jump into one of these tissues and look at the distribution of sex and exercise and control group. So here we have our 78 tissues, and we want to know how is sex divided? We see it's divided perfectly. It's totally orthogonal. And we can look at our control and exercise groups, and they're divided really quite well as well. They're not totally orthogonal, but the later exercise groups, 7, 24, and 48, have a lesser degree of rats in them. So 39 females, 39 males, and anywhere from 10 to 6 rats per experimental group. So let's look at an example of what this study was originally trying to do, measure the acute exercise response. This is a gene in kidney called Arnt. Now, if you are familiar with Arnt, you'll know that this gene is associated with circadian rhythm. And so what we'd like to do in MotorPack is model the acute exercise response with something, let's say, this is a hypothetical model, but something like this. Usually what we see is sort of the vast majority of the time, we see genes that are up or down regulated with an immediate response to exercise, and then they sort of taper off. The color scheme here is important too. It'll sort of help you to trace the experimental time points. So at 0 hour, we have yellow dots all the way to 48 where it gets brown, and we have an increasing gradient of darkness. And the same is true for the blues as time goes on for the control groups. Now there's a red flag here. I want you to look at where the normalized counts over time plot for the two control groups. These two control groups really haven't had much done to them. They've just been sacrificed at different times of the day. And so what you can see is the difference in their normalized gene counts is really quite considerable. And so there's a red flag here. Immediately on our radar, we're worried about circadian rhythm, the effective day when these samples were processed. So we're not just looking at one time scale here, not just this time scale of 0 to 48 hours. Instead, we have two time scales. It turns out that these samples were not processed all in the same order. They did not undergo exercise at the same time. And so our other time scale is the time of day. This is a busy slide. Bear with me. We're going to walk through it. In red, we have our samples that underwent exercise. They're in decreasing order from 48 to 24, all the way to 0 hours post-exercise. In blue, we have our control samples, again in decreasing order, 7 to 0 hour. We'll see two columns, start and end. Start represents the start of the end of the exercise bout, and end represents that time between exercise and sacrifice. This is visualized here. These red stars here represent the end of the exercise response, and the lines attached to those stars, and the very end of those lines, represent the time between exercise bout and sacrifice. Sacrifice, of course, being at the end of these lines. Now, in the control groups, they didn't undergo exercise, but these are just, let's just call those control events. A few things to notice here. First, those controls that we were comparing before, they're at opposite sides of the day. And this day is between 10 a.m. and about 6 p.m. So this is a normal working day. In addition, most of the time points associated with the acute exercise really fall in this 3-hour period between 11, 12, 13, and 14. There's two specialties here. You'll see dotted lines here. Those dotted lines mean that this time actually represents over a 24-hour period. So it's gone all the way around the clock and it's come back in one case, or it's gone all the way around the clock for the twice for the 48 hour. Okay, so we want to measure exercise and circadian effects, and now we're going to plot these both on hours post-exercise as we've seen, but also by the time of day. So the same data is used to disentangle these two effects, and it's modeled best on two different time frames. So we're shuffling and we're rescaling the x-axis. So again, if we look at these control groups that differ considerably, what we can see is that, well, they differ, but they differ on different ends of this workday. So is it the time of day? Is there a circadian effect that's building up and changing the gene expression of these control groups? That may be for the control groups, but what's really sort of troubling is to look at exercise group 7. We see that right here. Is the expression from exercise group 7 also influenced by when it was collected in the time of day, or does it truly respond to the acute exercise bout? So in measuring these two effects, we're suspicious. We think circadian patterns may interfere with our detection of the exercise response. We know it's going to be difficult to disentangle these two effects. One of the big reasons is our lack of samples. The lack of parameters we can use in our models. We're really limited. We sort of have to get savvy here. We've only got 7 sampling points for the exercise groups. We've got 2 for the control groups. We want to know which effect is dominant. One thing we want to avoid here is the binary yes or no answer. Is this data set contaminated by circadian rhythm? Yes or no? We want to avoid that. Are certain tissues contaminated by effects from circadian rhythm? Instead, what we want to look at is every gene in every tissue, not in a binary response, but we want to quantify that response. How much of that gene is influenced or modeled better to the circadian model versus the exercise model? So we're going to model the contribution of circadian and exercise effect. We're going to quantify the difference, we're going to compare the percent variance explained from these two effects. And therefore, what we're actually doing is we're taking this sort of observational data and we're performing experiments. We're going to summarize those results in every gene across 15 tissues, and then we'll sort of come back and we'll take a bird's eye view. And hopefully, we're going to be able to differentiate circadian rhythm and response to exercise. We're going to take 2 strategies. This is our first strategy. We're working with RNAseq data, so we're going to very briefly run through the RNAseq QC, and I apologize for that. I know that we could spend a whole session just talking about QC, and it is really important. It lays the foundation. But we want this talk really to focus on the modeling itself. So we're going to jump into modeling after QC, and then we're going to look at the percent variance explained from each of these models. And at this point, these models are separate. So let's jump into that QC. So we decided to perform variant stabilizing transformation. We looked at a few different transformation techniques. I got to admit, this was my first project when I joined as a postdoc. What I know now, I'm not sure I would choose variant stabilizing transformation. I would look at other transformation techniques with much more rigor. You see variant stabilizing transformation when you think of DEC2. We're not even doing a differential gene expression analysis. Now, we looked at other transformation techniques and we think, we thought this was our best choice. But I do want to point out that this could be done with more rigor. And so when we come back to it, we'll look at different transformation techniques, and we'll look at them not only at the finish line, but all those little steps along the way with many QC plots to really understand how the data responds to transformation. With that said, I think we're okay with VST transformation for the big picture of this talk. What we have here is a box plot of our of 78 samples. And we can see that at least their medians are really quite comparable. These are normalized counts on our y-axis. And if we just take two samples, let's say we take sample 1, we take sample 2, what we see is that they're really quite comparable. By comparable, I mean if we had a line going through here, that line would be at 0 and 0 and have a slope of 1. So many of these percent variance explained might equal each other or are really quite heavily correlated. Now, some genes have expression of 0. One thing VST, quite a majority of them do, and they all line up here. What VST does is it actually shoots them up to 5. Let's talk about sample processing. For all 15 tissues, this is a bird's eye view. We started for the most part with 78 tissues. And we have 3 major decisions that we had to consider: sex verification, batch adjustment or covariate adjustment, and outlier removal. Sex verification, there was one reciprocal pair in hypothalamus. They were mislabeled by their sex. We could tell it, we could see this from their sex genes, and so we just switched their labels. With batch adjustment, for about half the tissues, we saw that sex was really driving the variance, especially on, let's say, PCA. And so we just centered those samples by sex and brought them together, because what we're really concerned with is the experimental groups and circadian rhythm. Then we performed outlier removal. Across these tissues, we removed 18 samples, and that left us with 1074 samples in total. So here's a PCA after all of that. And I just want to sort of take you on a grand tour here. We're going to start up here with control 0 hour or control IPE immediately post exercise. We're going to move over to exercise 0 hour, 0.5, 1, 4, 7. By the time we get to exercise 7, it pairs up with control 7. And when we go back to that 0 hour, what we see is that it pairs up with the 24 and 48 hour exercise response. And so we get this sort of grand tour, if you will, visualized with this ellipse. One thing I'd like you to focus on, we're going to look at all these tissues in a second, is the difference between our controls. Those are going to be in blue. And I've highlighted that, excuse me, I've connected them with a dark blue dot. So when we look at, can the audience see this all right? Yeah. When we look at all these different tissues, some of them show these circular patterns really quite nicely. And these tissues are the ones you would expect to have a response to acute exercise: adrenal, gastro, heart, lung, liver, kidney. Kidney's also blown up here. And some of them not, but we force the ellipses in there, or ellipticals in there, and then we show this distance between our different control groups. And this can sort of give us an idea of, with how well they fit the ellipse, if they are really responding well to acute exercise. And then with regard to the length of the dark blue line here, if circadian rhythm is having an effect. This is really just a superficial view of it. It's just to put it on our radar for certain tissues. So let's jump into the modeling. We're going to try 2 separate models, one for circadian rhythm and one for the acute exercise response. So let's jump into model 1. This is our model for acute exercise response. Believe it or not, we're using a polynomial model. This has 5 parameters. It's a 4-degree polynomial model. We're modeling normalized gene expression, and we are using hours post-exercise, but for our X, but we've square rooted those. One of the reasons we square root it is because it's a 4-degree polynomial, and we have hours post-exercise that go all the way up to 48 hours. One of the downsides to such a model is that these later terms, 48 to the 4th degree, is really going to steal a lot of the model, dictate the model's behavior. It might overfit on some of these latter points. On our x-axis, well, I should say one of the perks of the model. One of the perks of the model is it's incredibly flexible. Now, you can't draw an elephant with this model, but it does allow us the flexibility to capture the very diverse responses we're seeing in these exercise genes. So on our x-axis, we have hours post-exercise. We're using our exercise cohorts, but only one of those control groups. We're leaving one out almost as a test sample. And it doesn't really serve, as far as I can see, any use in performing the exercise model. You'll notice the same 7 time points, but this control 0 hour, actually we moved it back to -1. And that's because it was sacrificed just before the exercise event. That exercise event was a 30-minute period, and that really helps anchor the model. When we visualize this model again, you'll see why we made this modeling decision. The model 2 is a model for circadian rhythm. This is a model with 3 parameters: baseline, magnitude, and phase. It is assumed to have a 24-hour period, and an equivalent function is right here, where we are modeling the normalized gene expression, and we have our sine and our cosine terms. In xc right here, we've got 2 pi times the time of day. Time of day is on a scale of 24 hours divided by 24 hours. And our x-axis is time of day. We're using, of course, we're going to use the exact same models, control 0 hour and the exercise cohorts, but our x-axis changes. And so our order is that the order of the hours in the work, 10 a.m. to 1800 or 6 p.m. All right, so let's jump into this. Let's look at an example of a gene that was modeled fairly well by the acute exercise response model. So this is PDK4 in kidney. This is a gene known to play a role in acute exercise response. So here we have our 2 models. On the left, we have our acute exercise response model. On the right, we have our circadian model. Here you can see the annotation of our gene groups. You'll see that control 0 hour that we push back sort of as an anchor. Underneath, you'll see statistics. PVE stands for percent variance explained, R squared, and p-value. We can see that PDK4 actually fits both models well enough, but it fits the acute exercise model better. But still, we have a problem here because it fits both models. It's certainly not mutually exclusive. And then we didn't use control 7 hour in our model, and that's right here, but we sort of throw it in there just as a check and balance, as a test sample. Does it really jump in and fit our model? Here it sort of fits on the line, but one might argue it fits on that line as well. So let's look at a gene on the other side of the spectrum. This is a master regulator of circadian rhythm in kidney. And very much the same in our exercise model and our circadian model, we see that this gene fits really quite well. And it fits really quite well because we have this confounding between the time of day and the hours post-exercise. It's really this strange little coincidence. So we have, let's just focus on the PVE here. We have high percent variance explained by both models. So we need a way of differentiating these. And then, of course, if we add the 7-hour control group, it could fit conceivably to either model. So let's look at the variance explained by these two separate models. Not surprisingly, they correlate really well. Here we've got a line from 0, 0 with a slope of 1. And so a lot of these PVEs from these two separate models might actually, believe it or not, equal each other. If they don't, they're darn near close. And so we just have a hairball. So we have a problem. A lot of these genes fit both models, and we need to differentiate them. We want to differentiate their effects. So one thing we can do is model jointly. Do the same thing and ask what's the distribution of percent variance explained. Except the only difference is now we're going to combine the model. We're going to put the exercise model in the first part of this combined model and the circadian aspect of the model in the second part of the model. And we're going to take those two parts in red and blue, and we're going to make them compete for the percent variance explained. All right, so let's look at those genes again. You've seen these before. This is nothing new. These are those independent models. They're simply up here for reference. We're going to fill in this area below, just to show you that they fit both models for Arnt-L, or Arnt-L fits both models. But here's where it gets interesting. When we compare the acute exercise model right here, it hugs the residual. This is the residual, this dotted line right here. We've got two different time scales here, so we can't put both curves on both graphs. But what we can do is examine each respective curve on its respective graph, and we can see immediately that the exercise model really is not modeling well. It's hugging the residual. Now, that's not the case for the circadian model. It's modeling really well. It's stealing that, it's explaining much more of that variance. In addition, let's take part of this combined model from the exercise part of the model, and let's calculate the Y, or in this case, the normalized gene counts. And let's plot them. And that's what we've done here. What we would like to see with these red dots is that they actually fit in to our groups. They don't. There's one exception, but that exception is on the residual. Instead, they just hug the residual, much like you see here. They're not capturing much of the variance. Now, on the contrary, we can do the same exercise, taking the circadian model, which we believe fits better, and we can plot those Y values just with that, with the end part of our model, the model associated with circadian rhythm. And what we see is that those points in blue here really jump into our experimental groups quite nicely. We can do the same thing for PDK4. Remember, this was a really a well-known exercise gene. Again, this is for reference. Here are separate models. They both fit relatively well, our exercise model and our circadian model. And then when we combine them, the story's not so clear, but you can still see the themes. You still see that the exercise effect model is following our major groups and modeling much more accurately than our circadian rhythm model. And then if we go through the same exercise and plot the points, we can see that the circadian models Y values really don't fall into the groups, where if we were to do the same thing for the exercise model, they fall much more closer to the groups. This one's not as clear cut. And so we need a way to visualize the percent variance explained in every tissue for every gene. This is what we had before. This was using these separate models, but really when we combine models, we create a, what is this, a 2D density map, where we have the log of the gene counts right here, what we can see is first, the majority of genes aren't fitting either model well at all. But a minority of the genes are modeling better to the circadian model, where the majority are really modeling better to the percent variance explained from the acute exercise part of that model. So we need a systematic way to sort of categorize these different, let's say, plots of real estate. And so we've done that. We've taken these plots and we've labeled them 5 different plots from 0, 1, 2, 3, and 4. So 1 would represent those genes that model better with the circadian rhythm aspect of our combined model, 4 with those of the acute exercise aspect of the model, and 2 and 3 are somewhere in between. Notice there's much more in 4. Notice a sort of more pronouncement over to 4. They come out a little bit further than 1. And then the cardinal sin of going over 100% variance explained does not occur in any of these genes. So let's look at every tissue, every gene and every tissue. So we've still got our 4 groups, 0, 1, 2, 3, and 4. And I've outlined 3 tissues here. These 3 tissues really seem to respond to the acute exercise response. And you can see them over in this heat map here. It's got all of our tissues here, and it's got our different gene groups, 0, 2, 3, and 4. And we've annotated them as a heat map. We've not annotated group 0 just because the vast majority of genes are actually in group 0. And you can see that it's adrenal, gastro, and ovaries that have the highest frequency of genes that fit in this group 4. In addition, you see a sort of behavior, you see a sort of waning over of these genes into group 4. And so this is a really powerful visualization. It helps you see what tissues are really responding to the acute exercise response, and then also sort of the degree of which circadian genes that model well with circadian rhythm might be adulterating it or pulling it over to the left-hand side. So we can see the frequency of those genes here and then here in all of our different groups. This is a 3D bar plot. It represents the exact same data here as well. So in 4, where you see these pronounced groups right here, you can also see them with these yellow little buildings or bars here. But notice how the genes in group 1 are just incredibly small, and then in 2, they're a little bit bigger. 3, they're bigger. 4, yeah, they're considerably bigger. Remember, our primary variable of interest is measuring the acute exercise response. So now I'm going to jump into really just a quick and dirty pathway enrichment analysis. What we're going to do is we're going to take KEGG mouse genes from 2019 and just run them through Enrichme and ask what pathways show up for enrichment. Again, we're using mouse in the annotation, but really we're working with rat. I'm going to divide that up by their gene groups, 1, 2, 3, and 4, except I'm going to pool them across tissues. So that, again, this is really quick and dirty. So here are our results. Here we have our negative log FDR. Our odds ratio is represented by the size of the circle. We have those enriched pathways. We filtered out some of those pathways that we thought were not very important. And we have our different gene groups annotated by their color. Remember, group 1 was that group that modeled better with the circadian aspect of the combined model, and 4 on the other side of the spectrum. Not surprisingly, 1 is enriched for circadian rhythm. What's really important is 2, 3, and 4 are not enriched for circadian rhythm. However, that doesn't mean that circadian rhythm genes aren't finding their way into 2, 3, and 4. It's just that they don't make, there's not enough of them to garner an enrichment. 2, we don't see much, but 3 and 4, we see, as expected, a lot of the pathways, mTOR, PIK3AKT, MAPK, that are really associated with exercise response. So then we sort of wanted to flip this on its head. Now we want to take the KEGG genes from mouse that are specific to circadian rhythm. There were about 30 of them, and we wanted to ask, okay, well, where do they fit in these different groups? Remember, 1 is the group that models best with circadian rhythm, 4, of course, with acute exercise response. We see the majority fit as expected. You can see that in these blue bars. In group 1, they model best to circadian rhythm. Now, here's something that's interesting. Some of these genes show up in groups 3 and 4. So it could be that there's some circadian rhythm genes that are responding to acute exercise. And that's really interesting. That actually may have, depending on which genes these are, if these are master regulators of circadian rhythm, and this is a pipe dream here, but let's say these are the ones that are going to make you really tired. We haven't investigated these, but you go on a run, you do some sort of exercise, and you adjust your circadian rhythms, and maybe you're not so tired later in the day. That's totally a wild hypothesis in a pipe dream at this point, but it would be cool. So anyway, we're coming to the end of this talk. What we've shown is joint modeling of circadian rhythm and exercise response. We did this to address the deficiency in the exercise design. We showed a global summary of relative effects. The exercise effect was really stronger for most genes and most tissues from our experiments and the data, but this varied, of course, by tissue. Most of those hallmark circadian genes were found in groups 1 and 2. However, interesting enough, a handful of them are found in 3 and 4 and actually model better to the acute exercise response. So are these circadian genes that respond to exercise? And we'd like to continue this analysis on different omics. We mentioned we have metabolomics, we have proteomics. We are transitioning over into the metabolomics, and we're really intrigued with the patterns we see here. Maybe they'll be similar but different, I don't know. And of course, we need to improve our modeling approach. We have a polynomial model, and that should be investigated more. We should run simulations on that model and see does it really model well in the space that we're trying to model it in. So more rigor is needed with this analysis. With that said, I just want to thank the people who helped in this endeavor, Jaiyu Yama, June Lee. Really, this analysis, the ideas, the concepts were really driven by June. I'm just the analyst. I joined his lab to learn, and Chuck Durant as well. So thank you very much for your time, and I'm happy to answer any questions.
If anyone has questions in the room, of course, feel free to speak up, and online you can put the questions into the chat box, or you can unmute yourself and ask.
Okay, I had asked the question.
Yeah.
So you're talking about some of the circadian rhythms. Did you mention, was the one example PDK4, which is a circadian rhythm gene, but it did seem like it was modeled better by acute exercise. So could that have been one of your circadian rhythm genes that were jumping into group 4 potentially?
So if I said that, I misspoke. PDK4 is a gene that's known to model well with the acute exercise response.
Okay.
And in our separate models, it does actually model quite well with the circadian rhythm model. But then when we combine models, then what we see is that really the acute exercise response takes over. That said, it seems to have a minor influence from circadian rhythm. So this could be an exercise gene that is being influenced by circadian rhythm. Whether this gene is known as a circadian rhythm gene, I don't think it is. I could be wrong, and if I am, please somebody speak up and correct me. But it is known as a strong responder to acute exercise.
At the moment, there are no questions in the chat, but again, if you're online and you have a question, feel free to enter it in the chat box or just unmute yourself and ask. We'll give it a couple of minutes if there are no more questions in the room, just in case anyone wants to type something in the chat box.
Sure.
If there are more questions in the room, please feel free to ask them.
It must have been an exhilarating.
You explained everything so clearly. All right, so I'm not seeing anything in the chat box. I'm not hearing any more questions in the room or unmuted microphones. So I think that we can give a speaker another round of applause. And hope everyone has a good rest of their day.
All right, thanks, everybody.